Hi everyone, so in this video I'm going to talk about eukaryotic transcription and all the important sequences that are involved in transcription. So I'm just going to draw out like our basic sort of DNA, and I'm going to name that one 3', 5', 5', 3'. So it's just some sequence of DNA, and we're just noting the directionality because that's important in defining where the template strand is and where the coding strand is. But one thing that you wouldn't be able to determine in this case is because I haven't given you a promoter, you don't actually know which way polymerase would go, right? So it could bind to this strand and then go that way, or it could bind to this strand and go that way. So in eukaryotes, unlike in prokaryotes, we see that there's this special sequence called the Tata box that is in the promoter. So it's really similar to the -10 box in bacteria, but basically there'd be this sequence. Actually, before I do that, I'm gonna redraw this. Okay, it'll become more apparent in a sec, but I'm going to draw this as being the promoter. And so somewhere within the promoter, there's this Tata box. So I'll just draw it like this, but just imagine the promoter as being a little bit bigger. But this Tata box is really key because what it's going to recruit is the Tata binding protein. So it's just some protein blob that'll bind to this Tata box. And then other proteins will come and bind around it. So you'll get like a bunch of things sort of binding. Okay, great. Yeah, and those are known as general transcription factors. So essentially, all of these together, so the Tata binding protein has found the Tata box, it's bind to it, and then the general transcription factors will assemble around it. So all those together play the same role as the sigma factor that we saw in prokaryotes. And so that's what will eventually recruit RNA polymerase. So then RNA polymerase will come and sort of bind somewhere here. It doesn't really matter like the details of that, but just to get a sense of sort of what's acting as what in this situation. So the Tata binding protein, the general transcription factors are like a sigma factor, and then the RNA polymerase sort of is recruited in the same way. But as I drew sort of this like slashes here to represent that there's sort of a break in DNA, and that was to show that there's actually sequences far upstream from the promoter, like pretty far away, that are known as enhancer sequences. So we didn't really talk a lot about these details, but it's just important to be aware that they do exist. And essentially, they're a sequence that's recognized by enhancers, and these enhancers will bind to these sequences and basically just like enhance transcription. So I don't really think you need to know more than that, but just to keep in mind that eukaryotes are a little bit more complicated in terms of the regulation of transcription, and so just be aware that those sequences do exist. So once RNA polymerase has been recruited, it'll start transcribing, and so I actually didn't draw these very long, but let's say these were longer. So in eukaryotes, we actually call the first, like the +1 site in prokaryotes, we call that the transcription start site. In eukaryotes, but for the purposes of this class, I think we've talked about in class how you can basically use them, like you can just call them both the +1 site because they really are the same general thing. So yeah, but just like a note that it really is called the transcription start site, but we can just call it the +1 site in this case. And so once the RNA polymerase reaches the transcription start site or the +1 site, that's when the first RNA base is added to make the RNA, and then it'll keep reading along the strand. It'll keep going until it reaches the terminator sequence. So terminators in eukaryotes are a little bit more complicated than what we saw in prokaryotes, but basically what's important is that the termination sequence is also transcribed, but there are multiple things that can sort of happen after this. So really just you need to know that there's a terminator sequence, it's transcribed like it is in prokaryotes, and that somehow sends a signal to RNA polymerase to stop transcribing. So hopefully that gives you like a general idea of what's going on. I guess the other thing that I'll mention about prokaryotes is that, or sorry, eukaryotes is what we see is in our resulting mRNA strand. This is mRNA. We have this special thing called RNA processing, right? That doesn't happen in prokaryotes because in prokaryotes, as soon as the mRNA is even starting to be made, ribosomes can already bind to it and start translation. So there's actually this really important part of eukaryotic processing that happens, and that includes things like, here I'll use a different color. So let's say, oh, I probably want to draw that more clearly. Okay, so let's say this little segment, so these little yellow segments, I'm going to call my introns. So the introns, like we discussed, are sort of these segments that are going to be cut out. So these will be removed during processing. The other thing that we mentioned was how the exons, so actually the regions in between the introns, some of those can also be removed. And this is a really important factor that we don't see in prokaryotes because in eukaryotic cells, the mRNA transcript can actually produce multiple proteins depending on what exons are removed on top of introns. So we saw the example of insulin of how in one case we had no exons removed, and in the other case we had one exon that was removed, and that changed the sort of the functionality of the receptor to insulin. So yeah, so that's really important to realize is that prokaryotes don't have this special ability to sort of have one mRNA that can make different proteins and sort of have different functionalities. So that's something that's really unique about eukaryotes and about the fact that they can do this processing. So the other things that are added are a 5' cap and a poly A tail. And so essentially they increase the stability of the mRNA and they prevent rapid degradation because when this mRNA transcript goes out of the nucleus, there are factors in the cytoplasm that can actually lead to the degradation of the mRNA. So the 5' cap and the poly A tail sort of act like a toque and a scarf, you know, sort of protecting the mRNA from the elements outside of the nucleus. And the cap is especially important because this is where ribosome binding occurs. So that's really one important factor that the cap has over the poly A tail. So hopefully this was helpful in creating a picture of what's going on in eukaryotes versus in prokaryotes. Yeah, and I hope to see you in another video.